All right, everybody, thanks for joining us here. Glad you could make it back, even though we had that special seminar last week. So put it on your calendar. March 15th is the next seminar. Mehmet Toner from Harvard will be here. Extremely excited about that one. And then we've got a speaker for April and May lined up. Also, both excellent speakers in the area of optofluidics. But today we're very lucky to have a very nice guy and great researcher. Taher Saif comes to us from Illinois, Urbana-Champaign. So he started out as a civil engineer, got an undergraduate degree in civil engineering at University from Bangladesh University, and then a master's degree from Washington State. Then became a mathematician, degree in applied and theoretical math, theoretical and applied mechanics. I see. Okay, from Cornell,
Yes.
1993, and then became an electrical engineer. So it's a postdoc at the nanofab facility in Cornell. And then finally what's left, mechanical engineering. So professor of mechanical engineering in 1997 at UIUC. He's now a full professor. And I think you'll see from his presentation here that this guy's really a bioengineer. So and everything else, finally, he found what works. So, with no further ado, we look forward to your talk.
Okay. So during the lunchtime, I was warned that this crowd hammers speakers. So they told me to have enough food so they can survive. Actually, I have gone to quite a few labs this morning and afternoon, and I'm so humbled that I don't know how much I have to contribute, because most of the things that I have seen is really very enlightening. My talk would be may appear to be too trivial to some of you or many of you. Okay, so I have what I have. No time to change. So what I will talk about is more of like some of the more recent works. In fact, the way I will be talking is the first time I'm talking outside my campus or outside my students on this theme. So when Reza asked me to give a talk here some time back, I was thinking that this is a systems institute for systems research, where all of you think in a systems-oriented way, and I tend to look at things at a single component point of view. But then when I look at the website and look at the people working here, I begin to really think in terms of systems, because systems probably have a very broad definition. And the way I would cast the talk, or some of the result that you will see is actually very much driven as a systems. If you think of the human body, if you think of the animal kingdom, if you think of any living system, each one of us consists of trillions of cells. Just the brain itself consists of about 3 trillion cells. In any organ consists of billions of cells. If you think of mechanical systems, electrical systems, they have much fewer components that drive the system. If you think of a car, basically it's an engine that is run by a few pistons, and once the engine starts, that drives the other things. If you think of the cardiac muscle, it consists of billions of cells. Each one is an actuator. It's like the whole car is made of thousands of motors. And yet it works in an enormous synchrony. So if you think of the evolution in biological systems, it has to be an enormous synchrony between enormous number of components that have to have precise timing, unless the system would not work. The system that we are familiar with, the engineered system, has far less, few orders of magnitude less components to work so that the system works as a whole. But in biological systems, just to maintain homeostasis, just to maintain the temperature of the body, needs enormous coordination between these trillions of individual cells. So if I have one video which I collected from the website, from probably one of the YouTube videos. So this is the beginning of the embryogenesis of a frog. This is an African frog, and you can see many of these videos. So you can see again that if I play back, See, I don't know how to play back. Okay. So the two cells divide to 4, to 8, and then keep dividing. But if you notice that there's an enormous synchrony between the division rate, the timing of the division. Okay. And if you progress in the embryogenesis stage, which is the next video, which is, I have to come here. And then again, when the gastrosystem is being formed, there's a huge amount of mass transfer, coordinated movement of these thousands and thousands of cells by the time the organ is approaching towards the end of embryogenesis. So there has to be a fundamental synchrony, and in this case, what I mean by synchrony is the precise timing of events of many, many components that creates this system. So I have a hypothesis which has, if you look at the biological systems, you notice biological literature, you see that if the question of synchrony is arised at all, it goes back to the question of genetic response or biochemical stimulation. And there is, I find that there has to be something more than just biochemical signaling, because this long-range synchrony has to have some long-range stimulation, has to have some other means by which this long-range synchrony is being achieved. So I propose that if I have a rope here and somebody else is having a rope on the other side of the room, and if I pull the rope, the other person feels it right away. So this long-range communication happens right away, and that's mechanical. The speed by which this communication happens is limited by the wave speeds of information transfer in solids or in fluids. So if the moment I talk, you hear from the other side of the room, is because the acoustic wave transfer happens at hundreds of meters per second. So it's a mechanical communication. The rarefaction and pulsation of the air that goes through happens very, very quickly. So to make the point, I will show one experiment, which is almost an elementary experiment. And this will probably begin to show why I believe that mechanical long-range interaction may be responsible for this synchrony. So this is an experiment where you have a 10 micron thick PDMS film and about 15 millimeters in diameter. So 10 microns thick, 1500 microns in, 15 millimeters in diameter, not 1.5, so 1.5 centimeter in diameter. So it's pretty large. So it's a very thin film. So all we did, we put cardiac cells, primary cardiac cells, on this film. So these cardiac cells, when they land, they all beat at different frequencies. So they don't know each other, they beat at different frequencies. But if you keep them, allowing them to beat, these cells do not divide, so these are primary cardiac cells. But after 3 days, if you take a look at this video, so this picture that you see here is a close-up view of this membrane somewhere over here. I don't remember where, but somewhere on this film. So this is occupying about, each cell is about 50 to 100 microns. So you're looking at about a few hundred microns on each side, but still much, much smaller compared to the overall size of this film. So if I look at any of this point, what you would see that each of these cells beat, and then what you would see also is whole image has dynamics, which means the whole film is moving back and forth. So let me show you this video. And I don't know whether the cell motion would be detectable from your distance. So you see this whole membrane is moving, and it doesn't move. And I have done this, I have tried this in my lab as well. So when I connect it to this to the projector, I think it cannot transfer the data fast enough. So let's see, we go back. So it plays just a little bit and then it stops. I don't know why. Maybe I'm lucky this time. Okay. So the whole membrane is moving large distance, but I don't know whether you have noticed each of the cells are also beating at the same frequency. Now wherever you look in this big film, any place randomly, you see that locally the cells are beating at the same frequency as the whole membrane is moving. The question is, how do these cells begin to coordinate in an astonishing way over distances that are hundreds and hundreds of microns away and arrive at this synchronous frequency? if you take a small piece out of it, just by mechanically cutting it out, what you see here is a small piece, and this piece just cut out from this membrane, and you will see the oscillation. Now this is really annoying. It stops in the middle. So what you will see is that this motion, I have speeded up 5 times, so it stops. Anyways, so the idea is, even if you cut it out, now it rolls itself because the cells are pulling, but after it gets rolled out, I have speeded up 5 times, so it is oscillating at the same frequency as the previous frequency that you have seen before. So if I were as a naive, if I thought that if I cut out this film, I would see this would beat at different times, different places, because cells are all over the place. Because it's very, very flexible, so cells could deform any way it wanted. But that doesn't happen. The whole thing beats in a synchronous, orderly way. It is interesting, if I could play it more, you would see this film, this whole cylinder is moving along this direction at about 400 microns per minute, because it's squeezing the fluid from one side to the other. So it's kind of swimming as well in the fluid. So this is an example where in an in vitro lab environment, this long-range communication has ended up having a synchrony. Okay? So my hypothesis here now is that this synchrony has been achieved by mechanical communication. Okay? So how do we how do we prove this? So first, I'd have to fight the reviewers, because they would say that no, each cell is giving some biochemical process, biochemical chemistry is coming out, so that diffuses to the next one and the next one, and they get perturbed by each other. Well, one way to get away with this argument is by making them on a soft substrate. So if you think of what could happen, what might be happening. So if I think of a soft substrate, 2D or 3D doesn't matter, after I have these cardiac cells. So when this one beats, this could make the other one interfere with its beating by 2 ways. One, it could deform the substrate around it. So if it shrinks, then the substrate gets pulled, and this one feels the pull of this cell, and it begins to react in response to this one. Or it can send a chemical which reaches over here and interferes with this beating. So there are 2 possibilities. Either way, they would be talking to each other, right? My proposition is, if it is diffusion based, then it's going to take long time, because the media is all around, whereas if it is mechanical, then it's direct. It just, when this gets compressed, the other gets pulled, and this is how they could get synchrony. So to prove that this could be mechanical alone, we did the following experiment. We take a single cardiac cell, or you plate the cardiac cells in very low density, so they're far apart from one another, and you bring a probe, a tungsten probe, which touches this gel substrate, which is soft, and by pushing it into the gel and then pulling it back and forth away from the cell. So this is the cell, this is the probe contacts the substrate and then moves out, and then moves away from the cell and then moves in. As a result, it is stretching the substrate back and forth. So the red lines are the probe motion back and forth. So this cell was initially sleeping. This is quiescent cell. This is typically what happens when you put the primary cardiac cells. They don't all of them they beat, just a few of them beat. So this is one of them which is not beating. So by moving back and forth, which is a red line, the cell initially didn't respond, but about 7-8 cycles of this substrate motion, the cell begins to beat. This is the blue lines. What does this mean? This tungsten probe for sure did not release any chemistry. This is an inert probe. The only thing it did is move the gel around it, which transfers to the cell, and the sleeping cell woke up, and the cell starts to beat. And it keeps beating even after the probe has been removed. When the probe was beating fast, it was trying to catch up with the probe motion, but then eventually when the probe was taken out, then it went back to some natural frequency, if you will. Okay. So this tells us that mechanical stimulation alone could trigger the dynamics of a sleeping cell. So then the question is, to what extent this could happen, and to what extent is this, how far does this cell see the probe, and what is the effect of the substrate? So I promise this is probably the only equation I would have in my talk, but I think this is important because if you if you think of what is going on, so if I have a cell which is like a dipole, which is creating the pull on both sides, so it's contracting as a cardiac cell is contracting. So it's contracting the substrate, and now if you look at 2 points away from this cell, so the first point moves inward, the second point also moves inward, but they don't move the same amount because this is closer than this one. So as a result, if you had a cell sitting over here, it would see that part of it is moving more than the backside. So the cell gets stretched. How much does this get stretched? Does this stretching happens? Depends on 2 parameters. One is it is inversely proportional to the stiffness of the gel. This is exact calculation. There is no assumption here, and it is inversely proportional to the distance between this dipole and these 2 points. So in other words, x is the distance between the dipole and this first point. So this would be x, and g is the elastic modulus of this medium, of the substrate. So for the higher the stiffness of this gel, the less is the crosstalk between 2 cells. Further they are, the less is the crosstalk. Okay? But this gives us an idea the exact quantitative nature of the crosstalk or the communication between the 2 cells. So if you plot this, it's a trivial thing to do. If you plot this, this gives you the cell the dipole, the gap. This is the x in the x-axis, and y-axis is the how much is the stretching of the cell. So if I have a dipole here, how much is the stretching of the cell? That's what this function is. That's exactly what I just showed you before, nothing new. But there's something interesting I would like to point out, which is how do I know now that if I do put these 2 cells, what is the effect of this gap between the 2 cells and the substrate in between? So the hypothesis that I'm making here is the communication between the 2 cells is mechanical. Okay? Second, if it is mechanical, then this is the way the communication should die out. In other words, for the cell goes away, I should see less and less effect. If the stiffness is higher, I should see less and less effect. So that's the hypothesis I want to test. Okay? So, and it just tells us that if I had a cell sitting there, if another cell is sitting there, this is the way they would be seeing each other or communicating each other. Okay. So the experiment that comes next, I could test this hypothesis from a statistical mechanics point of view. So if we put many cells, cardiac cells, and if I group the neighbors by their distances, and if I ask the question that if I watch those neighbors, the nearest neighbors, and watch how long they beat together. Now this is important, how long they beat together, not one cell beating, how long both of them beating together. Then the farther the cells are in this pair, the less would be the communication between them. So what I'm doing here is I am grouping the cells by the distance between them, and then asking the question that day 1 to day 4, how many of them beating together. So as an example, if I had 100 of these pairs that are 0 to 5 microns apart, and out of these 100 pairs, what percentage of them are beating together? That would be the percentage on the y-axis. So day 1, cells that are within 0 to 5 microns apart, this orange line gives you quite a bit of them are beating together. By the day, by day 4, how many of them are beating together? 100% of them are beating together. So what this tells us that further the cells are, the less and less percentage of them are beating together, and as you go more and more days, less and less of them are beating together. So the blue one, only the first, only within 10 microns apart, the cells are all beating together. None of them after 4 days are beating together anymore. However, if you look at day 3, you see again a decreasing trend as a function of distance, but by day 3, which is the reddish color, they are they are beating together, but less and less as distance go by. If you now increase the stiffness, so this was done on 1 kilopascal gel. So note that 0 to 10 microns, all 100% of them are beating together. But if you go to high stiffness, 47 kilopascal, which is 47 times more, which means that, remember, it was inversely proportional to the stiffness. At the 4th day, almost none of them are beating together. So if you go back to what we had said before in this relationship, that increasing the stiffness by 47 times would require that the cells be 47 times shorter from one another. So the distance had to be brought much, much closer to with respect to one another, because they go the same way. So what this experiment shows, that if I increase the stiffness, this beating together phenomena dies out almost, because they cannot see each other. And within a softer gels, where the communication can reach a little bit longer, the cell beating communication dies out as a function of distance. If you look at the previous plot where I had the communication theoretically, and if you look at the histogram pattern, you see this pattern is not too much different from the histogram that we get here. So the proposition here is stochastically or statistically, the histogram pattern or the probabilistic density function is like the density function that we obtained from this simple or elementary calculation that we did from here. So at least this shows that the beating of the two cells, so why they beat much, much longer time together? The reason is very simple. So if one of these cells begin to beat, then this one compresses or shrinks, it pulls the other one, as if like this is behaving like the probe that I had. So when this begins to shrink, this one gets stretched, and so this, if it is quiescent, it begins to beat. Now when this begins to beat, it influences the neighbor which started to beat in the first place. So none of them can get to sleep. And anyone going to sleep is being woken up by the neighbor, and they begin to synchronize, and their synchrony continues. So they cannot become quiescent. That's why on the 4th day, the nearby members are all beating in synchrony, but there is no more synchrony further away, because the effect dies out. So, yes?
But the fact that you've got more cells, doesn't that increase the distance because you have more pulling?
Right. So if this is like looking at the two cells,
Yeah.
but then you begin to bring into many body problem, right? So then it becomes a Green's function type effect. So I have one cell here, and then I'm affecting this one, and then a little bit further away there's another one. I have a slide on that one as well. But you see, the distance dependence dies out so quickly that if you're 40 microns away versus 50 microns away, the one that is 50 microns away sees much less than the previous one. But your point is well taken. So if I have a cell 40 microns over here, and another one 50 microns over there, so I first communicate with the nearest neighbor. This nearest neighbor then talks to the next one, and then it's a chain effect. So by 3rd or 4th day, you may have 1 billion cells. They're all talking to each other, like a chain reaction, right? And they begin to synchronize. There's no choice. In fact, I should emphasize, it is not only frequency synchronization, it's a phase lag to be 0. So it's that precise. It cannot be attributed to a natural frequency. One can argue that this was a natural frequency, though reach the frequency. No, you have reaching the 0 phase angle, right? Otherwise, the whole membrane would not be moving. It's like the same way that the Millennium Bridge failed, right? In London, when they opened the bridge. So everybody went, thousands of people went walking on the bridge, and then as they started to walk, then the bridge started to move a little bit, and then more it moved, people synchronized more, and it becomes even worse, right? And then they all stopped, and the bridge had to be closed. So it is a synchrony that, in fact, I need this analog to explain my next slide for the bridge synchrony. So the question is there a threshold for our nervous system to feel the bridge is moving? If the bridge were moved just a little bit, then we could not feel. So somehow there was a certain amount of this bridge oscillation when the nervous system could sense it, and then it feeds back, right? And then I move more in terms of the bridge, and that enhances, that gets more and more. Is there a threshold that is required for this synchrony to happen? That's a natural next question. So imagine there is such a threshold. Okay? I don't know what the threshold is. I can give you an empirical number, because when this experiment was done, we asked the same question, that when we had the probe experiment, we asked the question that if I move the probe back and forth, I can see how much the cell is stretching. So if I keep on reducing the amplitude of motion, in other words, amplitude of these red lines, then is there an amplitude below which the cell does not respond? So it turns out that unless I stretch the cell more than 2.5% of its original length, the cell does not care. So that's like the threshold, at least from 10 experiments that we have seen. So in some cases maybe 2%, some cases maybe 3%. So it's on the order of 2 to 3%. So if there is a threshold for this synchrony to happen, then I can imagine that on one hand, if I can think of a threshold map, which is if I have the gap between the cardiac cell and the elastic modulus of the medium, so for a given threshold, I could think of that there is a certain gap and there is a certain modulus for which I reach the threshold. So this curve may look complicated to you, maybe not, but it is coming from plotting this function. That's all I'm plotting. So I have a du, d delta, which is the relative stretch between these 2 points, and on the y-axis, I have taken f equals to 1, it doesn't matter, and the y-axis is this function. So g and x is what I'm varying, keeping delta, d delta constant. So then I get a function which is this. So for a given threshold, given d delta, I could plot a whole function, which means that if I choose to have this 2.5% strain, what is the gap I need between the two cells, and what is the elastic modulus of the media that I need to reach that threshold? If I increase the elastic modulus, then I lose this communication. To get back to the communication, I would have to reduce the gap between the two cells. So in other words, two friends talking to each other, if the stiffness goes up, I have to be closer to talk to each other. Or I guess you get the point, right? If the stiffness is increased, then I have to be closer to get back to the threshold. Threshold in the sense, in these two friends, what is the minimum audio signal I need to hear my friend? So if you believe in this, then all it means that if I have a system where the cells, as it is now, they're communicating. So for example, my membrane, if in case the membrane material is changed, for the same cell density, the membrane would not synchronize anymore, or it will take much longer time to synchronize, or not synchronize at all. Okay, so does it have any physiological relevance? So I started to look in literature and started to talk to cardiologists for a very different reason, but it was an intriguing sort of eye-opening experience, is that in patients where we have atrial fibrillation. So what is atrial fibrillation? Is we have two atrial chambers and two ventricles. So the blood comes into the atrium. It's like the atrium when people get together. So it's like the blood coming in the atrium. And then the atrium compresses the blood to the ventricle, and the ventricle pumps the blood into the whole body or to the brain. For the patients who have atrial fibrillation, this synchronous compression of this atrial wall falls apart. So the membrane begins to compress, but at the same time there's a local vibration in the atrial fibrillation. What that causes, what that results in, is a local vortices within the atrial chamber. So with this residence time of certain regions of the blood goes up, blood clots, the clotted blood goes into the ventricle, and that clotted blood gets pumped into the brain or the heart itself, and that clotted blood causes the stroke. So the people who have atrial fibrillation has 10 times more risk of either heart attack or stroke. Okay? So I started to talk to cardiologists, and the hypothesis is that, or the observation is that, the people with atrial fibrillation has much stiffer atrial wall. How much stiffer? So if you look at the histologies of patients with atrial fibrillation, what you see, this is the normal tissue. These are the bands of cardiomyocytes, or myotubes in this case. They're in between are the tissue, the tissue that separates them. The patients with atrial fibrillation, the cardio bands are still there, but what you have is a large scale of what they call fibrogenesis. In other words, some extra tissue being built up by fibroblasts, which are much stiffer. To what extent it is stiffer is when these atrial walls are dissected. For normal tissue, the atrial wall is so soft it folds onto itself. So after dissection, it cannot survive by itself. But the patient with atrial fibrillation, it becomes so hard, it looks like the eggshell. So they remain as it is. Is this a mechanism by which we can explain that this band cannot communicate with this band? So the hypothesis is very simple. If the two friends become further apart, they don't talk to each other. Or if the media becomes too stiff, they cannot talk to each other. In this case, it's happening both ways, or maybe at least one way, which is the gap between them, average gap between them has increased, and the media that has been interspersed by them has now become much, much stiffer, evidence being here. So either way, the mechanical communication between these two bands would fall apart or become significantly reduced, and as a result, you may see one band shrinking at a time when the other one has forgotten the neighbor, and then it starts to beat at some other time, creating this local type of flutter, in addition to the AV node signal that is coming, which is a neuronal signal that is coming. So I'm not saying it is all mechanical, because there is a certain neuronal signal that is coming that is controlling the heart. But at the same time, there could be finer tuning through this mechanical communication that helps to synchronize the overall beating. But nevertheless, these two phenomena happen here. The gap between them increases, and the stiffness of the intervening media significantly increases. That may cause the atrial fibrillation. In fact, the cardiologist that I talked to first, and when I asked how do we get atrial fibrillation regarding a patient, and he said, well, it becomes much stiffer. Why the stiffness is related to atrial fibrillation, of course, there's another issue. But this is one way. This might be one way to look into this. So again, the synchrony between these thousands or maybe millions of cardiac cells, each one is an actuator, each one is a motor of your car engine. They're all talking to each other in a very long-range way, giving rise to synchrony is the point that I'm trying to make. When do I supposed to stop?
Basically, when you gotta catch your cab, we're gonna get you.
Okay. Okay. All right. So this is one example of synchrony. I mentioned that in biology, I believe the synchrony happens through long-range 4 interactions in many different ways. I'll give you another example. This is in neuroscience, and many of you work in neuroscience here. And in most neurons, we have a single neuron cell, and it has a very long axon that meets with other neurons or other muscle cells, forming neuromuscular or neuron-neuron junction. This is what we call synapse. And at the end of the synapse, there are little vesicles. There are neurotransmitters. And when the action potential comes, these vesicles are released or exocytosed, and the neurotransmitters are captured on the other side, or that excite the other side, and the information is transferred. And now these neurotransmitter vesicles play a significant role in memory and learning, because the more a synapse is used, the more times we look at the letter A using the synapse, using few synapses. Eventually it remembers, and the synaptic strength, in other words, the amount of postsynaptic current due to a given action potential increases permanently because the synaptic strength goes up. I've talked too many things here in one sentence. And this was the basic thesis of Eric Kendall. So he defined memory in 2 ways, the short-term memory and the long-term memory. In short-term memory, when a synapse is used few times, then the postsynaptic current increases for a given. So let's say I'm using this 100 times. After using 100 times for the same action potential, the amount of postsynaptic current goes up and remains up for certain duration of time. He mentions 30 minutes to 90 minutes. So you're driving your car, you see a house, and you forget about it, by the end of the day or maybe a few hours later. But if you see a crash, if you see an accident, a traumatic experience, that does not go away. So that creates a permanent change of the postsynaptic current and maybe permanent rise of the number of vesicles at the presynaptic terminal. Okay? And he describes the mechanism by which the presynaptic vesicle density can go up. That's what gave him the Nobel Prize in 1992. So the basic idea of memory is that when a synapse is used, the brain morphology does not change, or maybe changing just a little bit, but what really changes is the synaptic strength. In other words, to what extent I can release neurotransmitters for the same action potential. So synaptic strength changes or increases if the synapse is used many, many times. And one of the possibilities is the number of neurotransmitters that can be released increases. So this is well understood phenomena in neuroscience. So what we were looking at is the fruit flies. So in the fruit flies, these are the axons. These are about 100 microns long. They are meeting muscles at the end, forming neuromuscular junctions. So you'd expect these vesicles, the neurotransmitter vesicles, being clustered at this end as these synapses are formed. So what is the role of force? And I said synchrony has to be there. So there is no point of forming a synapse without getting the neurotransmitters, because neurotransmitters are released from the synapse. If I form just a synapse but no neurotransmitters, then it's of no use. Or if neurotransmitters come 5 years after the synapse is formed, I would never learn ABCD, right? I would have perfectly good synapse. So the synchrony of this clustering and synapse formation has to be done right, on the right timing. Has to be synchronous. So I'll show you what happens when this axon is about to meet the muscle. So you think that when you talk about synchrony, things have to happen at the same time. And this is a remarkable example. So if you look at what happens to the muscle, and if this works out, so the muscles begin to protrude this structure, that finger-looking structures, as it is very, very close. In the same video, I cannot show both because they're differently labeled. So the synapse is very, very close. And the next one, what you will see is when the synapse is approaching the muscle, what the synapse is doing. This probably would not play either. With this amount of light, you don't see. Oh, it's not playing. So you probably can see the growth of the synapse. Is it working again?
Press play again until it goes.
No, see what happens? It plays halfway.
It's okay. Press play.
Yeah, each time it's
If you click the pointer, then it pauses.
Yeah. So each time you press it, it goes.
Okay, so you can see probably that the growth is happening. So schematically, so it's like when it is within about 10-20 microns away, that's when the muscles begin to shoot those finger-looking structures, and the synapse and these finger-looking, filopodia-looking structures come out from the synapse. So if you go back, so schematically, this is what happens at the 12th hour of embryogenesis. This synapse, or the growth cone, is like this, and the muscle is beginning to do this. And by 13th hour, they begin to meet. By 14th hour, handshaking is almost complete, but at the same time, the vesicle density begins to show up. By 16th hour, this handshaking is fully complete, vesicle density has been fully matured. And if you look at the vesicle density by fluorescence microscopy, you see the growth cone is here, muscle is very, very close, vesicle density almost 0. But when the handshaking is done, in about 2 hours later, the vesicle density has been completely filled up. So in this process where this handshaking is going on, somehow these neurotransmitter vesicles are getting accumulated and complete in these 2 hours. The funny thing is, in these 2 hours, something else also happens. And that's where the force connection comes. Now, so traditionally people knew that this vesicle clustering is probably done by some biochemical signaling process. The cell is sending them signals. maybe something is going on. But it is intriguing because, if you believe in the signaling process, biochemical signaling process, this is from this point to the central nervous system where the cell is living is 100 microns. This is about 300 nanometers. So biochemical signaling through this tiny channel going 100 nanometers inside is a very slow process. But there could be motor proteins. They may be moving things fast enough. I don't know. But this experiment will show that there's much faster communication lines, which is if you take the synapse that is, if you take the axon that is growing, and if you cut it by a laser axotomy about 50 microns away, then you see the vesicle clustering is gone. Okay? So the student first thought that, well, that's what is expected because no signaling is coming from the cell body. But then if you take this cut end and just pull it by a micropipette, you see the vesicle clustering is restored. Okay? So force us to do something with it. Okay? So the first hypothesis that you can make is we know when this process is happening, that's when this is happening too. So if force played a role in this case on a cut axon, then maybe when the axon is forming this connection, it was also building up the force. Okay? So how do we test that? Well, this is the data that before in an in vivo wild type, you see large vesicle clustering at the end. If you cut it off, the vesicle clustering goes down. If you pull it, vesicle clustering is restored compared to the unpulled cut axon. Okay. So first hypothesis is then the axons themselves must be under tension. But if that is the case, then we know this clustering is essential for memory and learning. So if this force is important for clustering by default, if the answer is yes, then our process of memory and learning must be integrated with some kind of forces within the axons. So how do you measure these tensions in live embryos, which are about half a millimeter in long and about 300 microns in wide? So we work with MEMS all the time. So this is a simple MEMS device, which is a serpentine spring. So the idea would be if you have a guitar string, and if I want to measure what is the tension in the guitar string, I'll just pluck it and measure the force in the stretch. And if the tension is high, then I will need a lot of force to stretch it. So you can measure the force, and by looking at how much the spring stretches. So that's what we did. We made a MEMS device with this serpentine spring. These are length is about 2 to 3 millimeters, and so on. This is a very soft spring. And the experiment is the following. So this is again one of the axons, and we will be pulling the axon by this MEMS probe, and with respect to a reference, you would see the gap change, which is the spring force. And what you see is this gap is increasing quite a bit as the tension is introduced, and you get a linear curve, and it does not intersect to the origin. In other words, before we touched it, this had a force of about 2 nanonewtons. So you can think that 2 nanonewtons is a very small force, but it's also very tiny. It's about 300 nanometers in diameter. So if you translated this force, this is on the order of 5 meters of water pressure. It's a pretty high force. So it looks like very linear, but there's a non-zero force at the end. So if you do quite a few of them, we get a histogram of these forces. So the force seems to be on the order of 5 nanonewtons in these fruit fly embryos. So if you look at again in a zoomed out view, what happens is when the axon is growing, it has not formed the neuromuscular junction yet. If you cut at that time, then you see there's a small gap that produced. But after neuromuscular junction is being formed, if you cut, you see a big gap, about 22% shortening. So if I take a rubber band, if I pull the rubber band and then I cut in the middle, then I would see a rubber band retracts quite a bit and quickly. But if the rubber band was very loose to start with, if I cut in the middle, it will just stay there. Cut gap will be very small. So if you don't believe in MEMS, or if you don't believe in measurements with serpentine springs, then at least you can look at these pictures. and if you see this gap is very small versus this gap is very big, it just tells you that prior to synaptogenesis, the force is very small. But just after synaptogenesis, the force is very big. So while the synaptogenesis is going on, vesicle clustering is building up, the force is building up. This force goes right away from this end to the cell body instantaneously because it's a direct force. Okay? So we looked at the literature, we find, I will just have one more thing before I wrap up, which is if you believe the force does matter, then the next question would be that if you cut it, force is 0, so the vesicle density goes down. If I increase the force, increase the stretch, do I also get vesicle accumulation? So in this case, an experiment is I pull the axon and look at the vesicle density. Basically, the bottom line is by pulling by about 10%, the vesicle density increases by 300%. So there's a tremendous sensitivity to the force that we apply on the axons that relates to vesicle density. So if I look back into the literature, you find that in neuroscience, people have been looking at tension in the axons in vitro. So if you culture neurons, you see these long, straight lines which indicates that they're in tension. If you cut at this end, they kind of become like a noodle. So it is evidence that there are tension. And in the neuroscience community, there have been literature published earlier that this tension remains in the brain. This was a famous paper by Van Essen. And what he proposed is that during the formation of the brain, when the synapses are formed, then the axons begin to shorten and build up the tension, which gives rise to improved circuitry, more efficient circuitry, and as a result, you get this folding in the cortical apex. Now, if the brain size is less than a certain volume, then we do not get this folding. So he looks at the entire species and finds that the animals with smaller brains do not have this folding, and above a certain size you get this folding. So it's like taking your kitchen sponge and compressing, and the sponge will fold. But if your sponge is very, very small, the sponge will just compress but not fold. So those who are in mechanical engineering, this is what we call buckling. Right? If it is very big, it buckles very easily. If it is very short, stubby column, it wouldn't buckle. So his argument is beyond a certain brain size, you get this folding below a certain brain size. But then the argument goes further that what else does this folding do in terms of hierarchy of cognitive thinking? So the point is, the idea is that tension is there. The existence of tension is known. I'm not saying anything new. The only thing, if at all, is the connection between this tension and accumulation of vesicles at the synapse. So it looks like there must be a good reason why this tension is being built up. So I had several communications with Van Essen, and it looks like this connection between this tension and vesicle accumulation sort of brings to light the fundamental role of tension. And through this tension, it's like a network of cables. If you perturb any one network, all the network would feel the force. So this long-range communication is achieved by this network tension. So what I have shown so far are just two very basic things. These are two examples, if you will, that supports the hypothesis that synchrony in development, synchrony in biological functions may be related to the tension. And I have shown examples that cardiac cell dynamics gets mutually coupled through long-range Fourier interaction simply by deforming the media in which they live. And if they're too far apart, then this communication becomes too weak, and they cannot talk to each other. If the media becomes too stiff, then also they cannot talk to each other. And it seems like this long-range neuronal forces where the synapse is formed with the junction with the muscle and the cell body is far away, it builds up the force to synchronize the early development of the synaptogenesis. If this force is taken away, this synchrony falls apart. The accumulation fails. So the synapse becomes useless. So I'll stop here and take questions.
So this, I think he's hands that up for a long time.
No,
Very
very-
anxious.
very-
Please.
Did you try the other experiment looking at it? So if you pull the other side of the axon that you cut, So if the information comes from the neuromuscular junction,
Mm-hmm.
and what you're claiming is that it should go to the central nervous system,
Mm-hmm.
you should actually pull the other cup and look where the vesicles are arriving. If the information comes from the neuromuscular junction, and then the process is happening with the neuromuscular junction, the information didn't go very far.
So you're asking whether I pulled the contralateral synapse, or
Yes, so your
so I have two synapses on both sides, right?
well,
So-
it's only, in this case, it's the neuromuscular junction synapse that is
Correct.
that is forming,
Correct.
right? And that's where things are happening.
Correct. Correct.
And what you said in the beginning is that the mechanical tension brings that information to the central nervous system.
I don't know that. All I'm saying is that if that force is not there, or if we perturb the building up of the force by either cutting the axon off, then the vesicle accumulation does not happen. In fact, this talk lends to two central questions. None of the answers I have. One is who generates the force? And second, what is the connection between force and vesicle accumulation? So what's the underlying mechanism by which this tension builds up? How does it interfere with clustering? I don't know either of the answers.
You had mentioned about the frequency.
Mm-hmm.
Do all of your cell populations beat with the same frequency?
Mm-hmm.
Does it depend on the stiffness of the membrane?
Ah, good question. You know, we changed the stiffness. This was done like 2 weeks ago. That's why I said I have not given this talk. This is the first time I'm giving this outside my students and my campus.
So please, they better hurry up because you're going to Maryland soon.
So we tried the next stiffness, which was 50 microns. And it turns out that the cells did not survive. I don't know, either because functionalization was not right, but I could not do a more thorough work on this PDMS membrane. So I asked my postdoc, why don't you go and see 50 microns? It will give more stiffness. And for some reason, the cells don't leave. So it has nothing to do with the stiffness. I think it has to do with some flaw in the experiment. Yeah.
A lot of people use a nonlinear viscoelastic model for the axon,
Mm-hmm.
and if that's correct, then you should have creep.
Yes, it is
So how
not
does that
correct.
fit into this?
No, that is not correct. The reason it is not correct, and I can show you another 20 slides on this one, is because in the viscoelastic, the classical Voigt-Maxwell's model that people have used,
Well, that's linear viscoelastic.
the linear viscoelasticity, the main reason it doesn't work is it does not account for the active force buildup in the axons. So what I did not show you is if you bring the muscle close to the central nervous system, in other words, if the length is 100 microns, and if you bring it to, let's say, 50 microns, then you get a large extra length. And what happens is the axons begin to shrink until it builds up the rest tension. If you bring it to 25 microns, it does exactly the same thing. So no matter what the length is you produce, it builds up, it builds up the force to that initial value, more or less initial value. By the same token, if you increase the length, the experiment that I showed you, by increasing the length, accumulation goes up. The force does not go up. Goes up quickly, and then it decays. But it does not give you a force response that you can get from a model that does not account for active processes. So if you look at the papers that were about 10 years ago, what they have added is a growth dashboard, what they call, on top of this viscoelastic model. So the idea was to include the effect of the growth, active growth of the axon as the development happens. So some kind of an active dashboard, growth dashboard, which responds to a certain threshold of the forces. So if the threshold exceeds, then the growth dashboard gets activated. And more and more cytoskeleton gets added to the system. If the force gets below a certain threshold, depolymerization happens and a back transport happens. So the force again goes up, builds up. So that is as far as it goes in terms of modeling. But I think just having a mechanical model without an active force generator and an active growth process, I don't think the model works.
So in your histological image of the fibrotic atria,
Uh-huh.
the striking thing is the lack of cellularity in the fibrotic one.
Mm-hmm.
And there are obviously larger distances
Correct.
between
Uh-huh.
the cardiomyocytes,
Uh-huh.
the muscle fibers.
Mm-hmm.
But those distances are filled with
Fibrotics, right.
tissue, which
Yeah.
I imagine would be quite stiff.
Correct. Right.
So wouldn't that transmit force reasonably well?
No, less. So it's like if I'm walking on a soft substrate, then when I bring my two legs closer, then the medium around me can deform easily. And that deformation is felt by my neighbor. If I'm walking on a hard substrate and I'm applying a force between my two legs, I do not deform the substrate. It is so stiff. So that's the reason why on a hard substrate, unless the two neighbors are very, very close, the communication is limited to itself. So whatever force I generate, I do not deform the media around me. So harder the substrate, less is the communication.
I agree if you get to really, really stiff.
Uh-huh.
But you think it's that?
It's just inversely proportional. So in other words, if I double the stiffness, the cell would have to be half as closer. So it's just 1-to-1. It's not quite 1-to-1, but at least theoretically, just for our understanding, it is like okay. So it is inversely proportional to G and to some extent inversely proportional to the X. It has few other terms there, but to first order. So if G goes up, then the amount of deformation between these 2 points goes down when a cell contracts. But if the substrate is very stiff, then these 2 points would not move with respect to one another. It's just all the forces were limited to within the cell itself. There's no deformation around.
So would that imply, though, that there's, as you get too soft, that breaks down? Because you can imagine the same analogy of something that's extremely soft, when you squeeze it, it just deforms locally.
It only says that it will deform, the deformation will propagate long, long distance away. That doesn't mean that it would stretch the cell above also. So it is a good point. So what is sort of missing in this calculation is how much the cell would stretch. So if the stiffness of the gel is much, much softer than the stiffness of the cell, so when the deformation of the substrate is going on like a fluid, the cell would not care because cell is much stiffer. So when I show this result, and the reason why I'm showing a solid line and dotted line is the dotted line comes when I have a cell. And the cell stiffness in this case is 4 kilopascal based on the literature. And this trend of the curve comes from this equation. The exact value of the curve comes from this finite element analysis. So this curve basically tells us the physics behind. The exact numerical value of how much a cell will feel the neighbor depends on the relative stiffness between this one, between the gel and the cell. So if the gel is very, very soft, but I'm very stiff, even though you are compressing the media, and all around this media is all getting stretched, but I wouldn't budge because I'm too stiff. So it's really the relative stiffness between these two. But the concept is the same.
Yes, and in this experiment where you have the different groups of cells, are they on a nurturing media?
Right. So that's right. This is the regular cell culture media for cardiac cells.
So,
but they would not divide because they are primary cardiac cells.
And did you do it? My question is on the mechanism of communication.
Mm-hmm.
Did you do an electrical study at the same time that you do the mechanical one?
No, I haven't done any electrical field analysis. People have done that before. It's typically known as mechanoelectric feedback. That goes back decades now. So if you take a layer of cells, if you apply electric field, then you can create this sheet type of beating. All of the cells beat with the same electric field, and you can create waves of propagation. So that's how the defibrillators work when there is a sudden cardiac arrest. So electric field is probably much better understood. And I made a comment that on top of this mechanical communication, there is always this AV node response that is signaling from the lower part of the heart that is triggered by the brain, as well as the pacemaker cells, right? And when they die, we use pacemakers, artificial pacemaker that send the same signal when they fail to send the signal. So electric signal is no question is inevitable. I'm not claiming it's all mechanical.
No,
Right.
It will be interesting to measure the levels.
Right. So we wanted to isolate the mechanical part as much as possible. That's why the idea of bringing a probe, which is purely mechanical.
You showed in the beginning the morphogenesis and the lip of the blastophore.
Mhm.
Has anyone looked? I have always believed that has got to be mechanical. And they always talk about these substances, and it doesn't make any sense.